I'm going to start this talk with a genetics lesson because some people may be very savvy in genetics and some people may know nothing about genetics. So we're going to start at an introduction to genetics, and then we'll talk about the genetics of the person. What I mean by that is the inherited genetics that you're born with, and we'll end with the genetics of the tumor, which is what happens in the tumor itself. So for talking about our genetics lesson, we're going to start talking about personalized and precision medicine. It's what got me to go to school to be a doctor and a physician scientist. We're all unique. We all have individual thumbprints that makes us different, and we all have individual DNA. Now, our DNA as humans is actually very similar. It's what makes us humans, but my DNA has slight differences from your DNA and your DNA. It's what makes us all unique individuals. And it was really in the 1990s when people thought we could really harness the DNA for a personalized medicine. And that's when the Human Genome Project really started. And the idea was, instead of treating everyone with the same disease the same way, we're going to look at a particular person's DNA and be able to treat them or predict risk of disease for that individual. And I remember this day. Some people remember when the man landed on the moon. I remember in February 2001 when the Nature and Science journal came out with the completed Human Genome Project. I was a graduate student in the middle of my MD PhD program, and this was awesome. So the idea, again, once we could sequence a person's DNA, then we can take everybody, and instead of giving everyone the same treatment where we know it won't work for a large percentage of people, we can instead look at the DNA or look at characteristics of a person and give the right treatment to the right person at the right time. And it's the idea that we're evolving from a one-size-fits-all system to a system of predictive, preventative, and precision care. It takes a lot of people to do all this. It takes bioinformaticians, mathematicians, technicians, computer scientists, biologists, medicine doctors, nurses, genetic counselors, and it takes ethicists. Why does it take ethicists? Anyone remember the movie Gattaca, the mid-1990s? I loved these actors, so that was my movie. But the real reason I bring it up is because it talked about the ethical side of knowing everybody's genome and knowing your DNA sequence. This movie, it was Hollywood, but it said you're already predestined based on your DNA to get this disease, that disease, marry this person, have this job. And that's kind of a scary thought. And so actually in 2008, there was a law that came out, and it's called the Genetic Information Non-Discrimination Act, or the GINA law. And this law protects individuals so that health insurance and employers cannot discriminate against anyone for having a certain genetic condition. And this is a really important law to have in place when we're dealing with genetic predisposition to cancer syndromes. So there are a lot of pros and cons for personalized medicine. I'm not going to go through all of them, but the pro is that we really think it can transform healthcare so that we can really give the right person the treatment and predict and prevent disease. Some of the cons is that it does cost a lot of money to do DNA sequencing and DNA testing, and there's those public policy issues which I talked about as well. But the ultimate goals really are to provide personalized medicine to prevent and treat complex diseases like neuroendocrine tumors, provide knowledge of disease risk, and individualized drug therapy. Okay, we're going to take a step back. We're just going to say, what is DNA? Some people really know this, but some people probably heard it once maybe in school, and that's it. So let's start there. DNA is our genetic code, determining the characteristics of every living thing. DNA looks like a twisted ladder, and I love this picture, but I'm kind of a DNA dork. The rungs of the ladder are made from 4 types of blocks that we call bases. And the bases are abbreviated by letters, A, T, C, and G. There's about 3 billion of these base pairs in our DNA in our human genome. And the sequence of which these bases occur, they matter because that labels the instructions for how our bodies are going to function. And so we can understand DNA this is an example of a section of DNA sequence. It's a lot of A, T, C's, and G's, but in fact, we break them into 3-letter words or codons, and these 3-letter words go together. They string along to make a sentence, and you can think about the sentence as the gene. So the gene is broken up of these DNA base pairs into 3 little words going along. So a gene is the part of the DNA that tells our cell how to make a specific protein. It does that by making a message first, and that message is RNA. And that RNA then gets translated into the protein, and the protein is the building block of our body. And we have tens of thousands of proteins to make our body function. But in order to have the proteins that our body needs to function, it's all based on our DNA sequence. So now we're going to talk about variations in DNA. We mentioned that we all have very similar DNA. It's what makes us humans, but we all have slight variations in our DNA. So when we talk about variations in our DNA, the way I think about it is that the vast majority of them have little impact on our health because it just makes us all different: brown hair, curly hair, straight hair, etc. I think of it as the American versus British way of spelling a word. Theater with an ER versus RE, labor with the U, without the U. The meaning of that word is the same, but it's a slight variation that we all have. On the other hand, we can have variations in our DNA that actually change the meaning. And if it changes the meaning, it causes a damaged protein that's not going to work properly. And so an example here is state. If you change this T to an L, you have slate. Or if you add an S in front of the word eat, it becomes seat. Very different meaning of those words, but a single letter variation. So the benign variant, where it's just the American versus British spelling, is called a benign variant. When it changes the meaning of the word, it's called a pathogenic variant or mutation. We're now trying to use the words pathogenic variant, but we'll use them interchangeably: mutation, pathogenic variant. Does that make sense so far? Okay. All right. So now we're going to talk about inherited pathogenic variants versus the pathogenic variants in the tumor itself. So when we talk about inherited mutations or pathogenic variants, we call those germline. You're born with this change in your DNA. It's in every single cell in your body. And we can test for that usually by a blood test because, again, the change is in every cell of your body, including your blood cells. So this is the test that leads to us figuring out if there's a predisposition for developing cancer. And we're going to talk about some of the net predisposition syndromes. When we talk about tumor mutations, we call that somatic mutations. What that just means is the DNA change was not there when you were born, but it happened by chance in a single cell in your body, and then that cell grows and proliferates and becomes a tumor. And we test for that only in the tumor because it's not in the rest of your body. It's specific to the tumor DNA change. And we sometimes want to know what DNA changes are in the tumor because it might help us find a treatment that will be specific for your tumor. Okay, so let's talk about the inherited genetics related to neuroendocrine tumors. We'll start with pancreatic neuroendocrine tumors. About 10 to 15% of PNETs are associated with genetic syndromes. And those syndromes are here, and we'll talk about them in a minute. But again, 10 to 15%, that means 85 to 90% of people with pancreatic neuroendocrine tumors don't have an inherited genetic cause. It just is sporadic. It happened by chance, okay? But 10 to 15%, there might be an inherited reason for having these tumors. For lung nets and for small intestine nets, they're rarely associated with genetic syndromes. They're almost always just sporadic as far as science knows right now. But if they do happen with a genetic syndrome, it's often associated with multiple endocrine neoplasia type 1, and we'll talk about that in a minute. Now, to me, the reason that caring for carcinoid changed to the NET RF is because we want to encompass all neuroendocrine tumors, not just GI tract typical or bronchial lung carcinoids, and that includes some other neuroendocrine tumors. I'm not going to talk about all of them because they're neuroendocrine tumors in the skin, in the breast, in the prostate, they're everywhere. But I'm going to talk about the most common in addition to the P-nets, lung nets, and GI nets, and one of those is pheochromocytoma paraganglioma. These are neuroendocrine tumors of the adrenal medulla and the extraadrenal ganglia, respectively. And different than the GI nets, a very high percentage, 35 to 40%, are associated with hereditary syndromes. In fact, it's the most common solid cancer to be associated with an inherited cause. Okay, lastly, I'm just going to mention medullary thyroid cancer. It's a neuroendocrine tumor in the thyroid. I mention it because you'll see when we talk about the syndromes that some of the syndromes patients can get neuroendocrine tumors here. It's about 25% of all medullary thyroid cancers that are associated with a hereditary cause, and that's multiple endocrine neoplasia type 2, and we'll talk about that in a minute as well. So let's review all these syndromes I just threw at you. We'll start with multiple endocrine neoplasia type 1, and that's because it's the most common genetic syndrome that's going to be associated with the pancreatic neuroendocrine tumors. It's actually pretty rare in and of itself. About 2.5 people per 100,000 have MEN1, and it's called multiple endocrine neoplasia type 1 because there are multiple endocrine glands with neoplasia or tumors. And it's really noted for the 3 Ps. These patients who have multiple endocrine neoplasia type 1 have a germline inherited mutation in the MEN1 gene, and they're at risk, not saying they will get, but they are at risk to get pituitary adenomas. The pituitary gland sits at the base of the brain. They're at risk for parathyroid hyperplasia and adenomas. That causes high calcium, kidney stones, and some other problems. And 30 to 70%, it's a wide number because it varies depending on the study, will get pancreatic neuroendocrine tumors. And there are often multiple pancreatic neuroendocrine tumors in the pancreas. Patients with MEN1 also are at risk for many other neuroendocrine tumors. GI neuroendocrine tumors, they can get adrenal tumors very commonly, but they're rarely the neuroendocrine type. They get Pheos less than 1%. But they can get neuroendocrine tumors in the thymus as well as the lung. The next syndrome that's associated with neuroendocrine tumors we're going to talk about is von Hippel-Lindau disease. This is caused by a mutation in the von Hippel-Lindau gene. It occurs in about 1 in 36,000 individuals, so a little more common than MEN1. And patients with VHL, they also are not destined to get every single one of these, but they're at risk for lots of different cancers and tumor types. Kidney cancer, hemangioblastomas, which are tumors in the nervous system, endolymphatic sac tumors, which are in the inner ear, and then about 17% of patients with VHL will get pancreatic neuroendocrine tumors, again, often multiple sites in the pancreas. And they're also at risk for pheochromocytomas, the adrenal neuroendocrine tumor. About 20% of patients will get Pheos, and they're often bilateral in both adrenal glands. The next syndrome is Neurofibromatosis Type 1. I actually did my PhD work in this disease, studying the neurofibromas, which are benign peripheral nerve tumors, not neuroendocrine tumors. But this is caused by mutations in the NF1 gene, occurs in 1 in 3,500 individuals. These are the characteristics that define NF1. But it turns out that although it's not part of the clinical criteria, patients who have this genetic syndrome are also at risk for neuroendocrine tumors. Specifically, pheochromocytomas, it really ranges. It used to be thought only 1% of patients with NF1 got Pheos, but now we think that it's much more common, probably closer to 13%. It's just no one was looking for them before. And then very rarely, there's also an association with pancreatic neuroendocrine tumors with this syndrome as well. Okay, we're moving on. I know I'm throwing a lot. We're going to tie it all back together afterwards. Multiple endocrine neoplasia type 2. So this is similar to multiple endocrine neoplasia type 1, just in the sense that they get lots of tumors in lots of different endocrine glands. But with MEN2, it's caused by a different gene. It's the mutations in the RET gene, occur in 1 in 40,000 people. And patients here almost everyone gets medullary thyroid cancer, again, neuroendocrine tumor of the thyroid glands. And about 50% will get the pheochromocytomas, but there's no risk that we know of the pancreatic neuroendocrine tumors or GI neuroendocrine tumors being associated here. Lastly, I'm going to spend a minute on the hereditary pheochromocytoma paraganglioma pheochromocytoma syndrome, either way. This syndrome is caused by mutations in actually 5 different genes, but they're all part of the same complex. They're called the SDH complex, or succinate dehydrogenase complex. It controls energy in our cells. And it turns out that the risk of developing Pheopara varies, and it's not that high for some of these. So there's a lot of people walking around with mutations in these genes who never develop disease, but a certain percentage do. SDHD, D for dad, because this gene and AF2 are paternally inherited. So if you inherit this gene from your mom, you won't get disease, but you're still at risk to pass it on to children. But if you inherit from your dad, you're at risk for Pheopara. And all of these genes are associated also with kidney cancers, GI stromal tumors, perhaps pituitary tumors, some mention of pancreatic neuroendocrine tumors with SDHD, but very, very low risk there. There's a bunch of other genes for Pheopara. Had to throw it in because it is my net. I'm not going to talk about these because they're really rare, but just to know Pheopara has many, many, many inherited genes associated with its risk. So I just talked to you about all these syndromes. So you might be saying, oh my gosh, should I be tested for any of these syndromes? And it really depends. So I told you that pancreatic neuroendocrine tumors, they're only inherited up to 15% of the time. So we don't recommend that everybody with a PNET be checked for one of these heritable conditions. But if you have multiple pancreatic neuroendocrine tumors, or you have another feature of one of these syndromes, then you should have genetic testing to see if you have an inherited cause of these. For lung nets and small intestine nets, they're very rarely inherited. So again, we don't recommend testing everyone who has that type of net unless you have other features of these syndromes. For Pheopara and medullary thyroid cancer, it's a bit different because the inherited rates are much, much higher. So every patient with a Pheopara and every patient with medullary thyroid cancer should have germline inherited genetic testing to see if they have a heritable cause. So why do we care? We care because if you know you have an inherited cause, it's going to help your doctor screen you for recurrence, for another primary neuroendocrine tumor, and for those other features of all these syndromes that I talked about. So it's important to know if you do have an inherited reason for your net developing. And also, if you with the net have an inherited cause, we want to test your blood relatives, because if they don't have the gene, they're not at increased risk for developing this type of cancer. If they do have that gene change, then they may be at risk, and we can screen them so we catch things early. NETs, as we mentioned earlier, takes a long time to get the right diagnosis, but if we know there's an inherited predisposition, an inherited possibility, we can screen people early and catch things early. So the question I get asked a lot is, but if I know I have this, does it mean that I'm automatically going to get a neuroendocrine tumor, or that I'm going to get another one of the tumors in each of these syndromes? And the answer is no. I tried to point out that percentage of people with each of those syndromes, and for some it's high, but some it's not that high. So it doesn't mean because you have a gene that you're automatically going to get disease. It just means you're at increased risk to potentially get disease. And one of the research things that I do is try to understand everyone who has the gene mutation. How do we predict who's going to get disease and who isn't? Can we find genetic reasons why some people are protected and never get disease even when they have this gene? Because maybe that could be a therapy to give to people who do have disease. All right, I'm going to spend the last couple minutes just talking about the tumor genetics. This is different than the inherited genetics. So remember, this is the DNA change that's not in your whole body. It's just in the tumor itself. And we can utilize that perhaps to understand better treatment options for that tumor. So it's interesting because across all these different NET types I talked to you about, there's actually some similarities in the mutations that form in the tumor. Earlier we heard about ATRX and DAX. It's in pancreatic neuroendocrine tumors, and our research found them in pheochromocytomas and paragangliomas too. So there's overlap with these mutations that occur in the tumors, and that might help us better understand why neuroendocrine tumors form and why sometimes they can metastasize. And it might be that there's possible drug targets. So you might have had a surgery or a biopsy where your oncologist or surgeon sends that tumor tissue off to have genetic testing. Foundation Medicine, for example, is one of those companies. There are others. And that's to look at what DNA changes happened in your tumor. Not your whole body, just your tumor. Because maybe by chance, there's a mutation in there that we have a specific drug target to, and that we can treat you and your tumor with a specific drug based on the mutations. Now, neuroendocrine tumors, they're often pretty indolent, which is a good thing. That's why I can live for so long with them. That also means that the tumor itself doesn't always have very many mutations in it. Again, a good thing because it's not very aggressive usually, but it makes it harder sometimes to identify these special targeted therapies when there's not a lot of mutations in there. But again, maybe not a bad thing. Okay, so personalized medicine, again, taking that tumor DNA and instead of giving everyone the same treatment, find out based on the tumor DNA which treatment is best for which patient. The right treatment, right dose, right patient, right time for the right outcome. So 2 slides on the future where I see we could go. For NETs specifically, we already kind of use targeted treatments and therapies. So PRRT, Lutathera, and MIBG, Azedra for pheochromocytomas and paragangliomas. These are specific targeted therapies to specific receptors in the tumors. So we have to make sure the tumor takes up these molecules, and that's why we do the gallium dotatate scan or an MIBG scan. And if the tumor takes up those molecules, we can treat with this type of treatment. But now I could go even farther, and maybe the future will let us know what genetic markers will help us determine if you're going to respond. So just because the tumor takes it up, a lot of people respond, but some don't. And maybe we could figure that out based on the genetic markers in the tumor. Another future direction is using the person's DNA. So now I'm going back to your germline DNA. I already told you if we know you have a germline inherited mutation in all the cells in your body, we're going to screen you a certain way to look for tumors and try to catch them early. But maybe, and this is a lot of my research, we can use that DNA to figure out to predict who's going to get disease and prevent it from occurring. So this could be the future of some of the genetics for NET tumors. So hopefully I haven't scared you away talking about all the genetics. We covered that DNA variants can be benign or pathogenic. We talked about that there's many genetic syndromes that predispose to neuroendocrine tumors, but that not everyone who has a NET is going to have a genetic syndrome. And I should say not everyone with the genetic syndrome DNA change is going to get a NET. And that hopefully tumor DNA mutations are going to provide more and more information about therapies and how to treat specifically each individual person. So thank you very much for your time, and I look forward to your questions at the question and answer session.